All right, welcome to Biology 1441. This is a lecture for the spring of 2020. I want to point out to you all that we don't have SI for Bio 2, but we do have in Lewis Science Center 155 graduate students who can tutor you in the subject of Bio 2. And I have arrows pointing out the TAs who are currently teaching Bio 2 who would be able to help you the most. You might be able to get questions answered by anyone on this list, but those with the arrows next to them indicate the times when Bio 1441 TAs are available. All right, we have several learning objectives for the chapter, and this video will not be the entire chapter. This is just to get you ready for the first lecture so we can spend some time working on some of these problems in class. So we should bear in mind that we just talked about Darwin and natural selection. We talked about the importance of variation, heritable variation, without which there can be no evolution. Okay, and we can bear in mind that variation has several sources. For example, sexual reproduction creates variation, meaning we recombine existing alleles in new ways thanks to meiosis. But it doesn't create brand new alleles. It creates new combinations of alleles. Now, mutations in cells that produce gametes can produce brand new alleles, and those can be for example, point mutations like we talked about in Bio 1. If you don't know what those are, you can look them up in your textbook. Point mutations often usually sort of no impact, which we might call neutral variation. So there's, like a silent mutation has no real change to the protein. It can also have a negative change if the protein changes shape, shifts function, doesn't work anymore. Or it could have a positive change. Perhaps this organism now is slightly more resistant to an antibiotic if it's a bacterium or something like that. So lots of possibilities thanks to small changes in the DNA. Now, bigger changes can also occur. For example, entire genes can be duplicated, and then one of those genes might mutate, creating, a different protein over time or whatever. But mutations typically are rare, but they do happen. But in organisms that divide really rapidly, like bacteria, mutations can actually accumulate in a population over just a few generations. And so what will happen is you have a gene, like here we have, the numbers here represent the number of base pairs in a gene. So from the beginning of the gene to the end, and we have exons and introns, remember that from Bio 1 hopefully, and we have different sites where there have been substitutions in base pairs, and a lot of those will be silent mutations. But then we also can get insertions accumulating, which will start to change the protein produced quite a bit perhaps. And substitutions, here we have a substitution in an area that's possibly resulting in a different amino acid being produced, right? So occasionally a substitution of base pairs will have an effect and change the amino acid in that location. And then insertions and deletions may also have quite a bit of an impact. So different kinds of mutations can affect the proteins produced. Also in Bio 1, we talked a lot about Punnett squares. Here's a real basic Punnett square that I'm sure you can solve quite readily. I ask you to pause the video and actually solve this Punnett square. We have big B, little b, and we have big B, little b, and they're crossing with each other. Now in Bio 2, we are going to look at a slightly different way of looking at this. For example, we have here an organism CRCW and CRCR. We're going to look at some traits that are codominant in Bio 2 a lot, just because they're easier to demonstrate what we're talking about. Codominant traits are traits, rather than having a trait that's one dominant, one recessive, they're both dominant, and they'll both be expressed if you have them both. For example, in this case, if you are a horse and you have CRCR, so C stands for the gene, the color gene, and R stands for which allele you have. So CR is the red color allele for that gene. CRCR would be your genotype, and that would be saying that you have two copies of the red allele. and your phenotype therefore would be red. But if you are CRCW, you would be red and white. So these are traits kind of like blood type in humans, where you can be type A, type B, or type AB, for example. Now, one thing I want to point out, you're going to need to work on making sure you understand the difference between genes, alleles, genotypes, phenotypes, okay? Because evolution is essentially a measurement of the change in allele or genotype frequencies over a period of time. So frequencies are just what percentage of the population is that allele, right? And I'll explain that better as we go. But we could have, in this case, purple flowers, white flowers, right? We have alleles. We've talked about that already in previous coursework, and you are expected as a bio major to carry forward that knowledge into this course and into future courses. So don't forget all of that terminology. Now, one thing we can say is that evolution is going to affect the gene pool, meaning first of all, bear in mind the gene pool are all of the genes in a population. And individuals, like my dog down here in the corner, they don't evolve. A dog, a single person, they can't evolve. I can't evolve. You cannot evolve. Only populations over time. Populations, if you remember, have a definite definition. That's a group of organisms that can interbreed with each other in a geographic area. Populations evolve over time. And this chapter focuses on what we might call microevolution, which is small scale or population level evolution. So changes in allele or possibly genotype frequencies as well over time. Now, a frequency, once again, is a proportion. For example, if 5 out of 10 individuals in a population have the CR allele, that would be a frequency of 0.5 or 50%. Okay, that's what we mean by frequency. 0.5 would indicate that 50% of the individuals in that population essentially have that allele. That's a rough definition, but we'll refine our definition as we go. Now, one thing to keep in mind, that allele frequencies, so if you look at the color allele in a population, and in that population 50% of that gene, that color gene, is red, then it's 0.5 is the frequency for that allele, so it's found 50% of the time. If evolution is not happening, at least not affecting that particular gene, then that gene frequency will be unchanged from generation to generation. Let's say it's 0.5. 50% of the color allele in this population is red. Then in the next generation, it'll still be 50%. And in 10 generations, it'll still be 50% if evolution is not affecting that particular gene. And this is known as, and can be calculated through the process of the Hardy Weinberg Equilibrium. Things will stay balanced. Things will stay in equilibrium if no evolution is happening. So the Hardy Weinberg Equilibrium is basically a null hypothesis. If you've taken stats, you know what that means. H sub 0, meaning there is no influence. Nothing is changing. We'll write some null hypotheses in lab. For example, if you are testing the effects of a drug on heart rate, the null hypothesis would be there will be no influence on heart rate by the drug. Okay, that's the null hypothesis. So what will the genotype frequencies be if a population is not evolving? So basically keep in mind that what we can do here is use this equation to answer that question. What will genotype frequencies be if that gene is not evolving? So if allele frequencies are not changing. And so what we can do is we can look at a population of organisms and begin to calculate this out to see if that population is evolving. So basically what you do is you calculate what genotype frequencies would be if that allele frequency or that gene is not changing, and you compare that to your null hypothesis. The null hypothesis states things won't change. You look to see if there was change. If there was change, then evolution is happening. So let's just look at an example. It's easier than talking about it the entire time. So you're going to be calculating these yourself, so I invite you to start working on it now. So here's a population of round organisms. They come in 3 colors, as you can see, 3 phenotypes, blue, yellow, and green. And there are two alleles here, color Y, CY, that's the yellow, and CB, which is blue. And if you have both blue and yellow, you come out green. Okay? So, what you would want to do, first of all, in this case, is you want to figure out how many organisms total there are. So there are 10, if I did my slide correctly. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 of these organisms. Okay, now because there are 10 organisms, how many alleles are there? Well, there are 20. Why? Because each organism has 2. CB, for example. There's 2 alleles per organism. So there's 20 total alleles in this population. All right, now out of those 20 possible total alleles, how many of them are CY and how many of them are CB? That's how you determine your allele frequencies. How many are CY and how many are CB? So if we look at this population, we can see that there's 1, 2, 3, 4, 5, 6 yellow alleles in the yellow organisms. All right, there's 6 of them there. And there's also 1, 2, 3, 4 yellow alleles in the green organisms. So that means out of the 20 total alleles, 10 of them are yellow. Well, 10 out of 20 is 50%. So we have an allele frequency for yellow of 0.5. And also, if you do the same thing for the blue allele, you also get a frequency of 0.5. 0.5. Okay? Now, let's also figure out the frequencies of our 3 genotypes. Out of these 10 organisms, how many are yellow? Well, 1, 2, 3. So how many are CY? 3 of them. So 0.3, right? How many are green? 1, 2, 3, 4. So 0.4 is the frequency of the heterozygous group. How many are blue? 1, 2, 3. Once again, 0.3. So we can figure out our allele frequencies, 0.5, 0.5, and we can figure out our genotype frequencies, 0.3, 0.4, and 0.3. Yeehaw, right? What's the point of all this? Well, we can tell if evolution is happening in this population by knowing this information. So we've already calculated this out. You can type this out or write this out yourself, fill in the actual data. But I will go ahead and project it here. We have 0.3, 0.4, 0.3 for genotype frequencies, and allele frequencies were 0.5, 0.5. All right, now the next step, the Hardy Weinberg Equilibrium. Here we see the equilibrium at the top of the screen, p squared plus 2pq plus q squared equals 1. P is a variable that stands for the frequency of one of the alleles. Q is the frequency of the other allele. Okay, so we'll just make P be the yellow allele and Q be the blue allele. Okay? All right. All you have to do now is stick those allele frequencies into this equation. So P squared, so remember those allele frequencies. Let's remember what those were. Let's step back one slide. 0.5 and 0.5 for both of those, okay? So you plug that in. P squared is 0.5 squared. That's what I wrote right here. Plus 2 times P times Q. That's 0.5 times 0.5 times 2 plus Q squared, which is once again 0.5 times 2. So now it says to simplify that equation. All right, I'm not telling you to solve it because we already know it equals 1. It will equal 1 every time if you set it up properly. Equaling 1 is not really important for solving this problem. It's just letting you know that you set it up right. Because 1 means 100% of the population. So if your values all work properly, it'll always equal 1. So now we simplify the equation. That's the important part. Not solving the equation, but simplifying it. So you take 0.5 squared. What is that? 0.25, right? So looking at the next slide, I've simplified the equation. 0.25 plus 0.5 plus 0.25 equals 1. Okay? So that is the solved, essentially, Hardy Weinberg equilibrium. Now, let's look at it. What does 0.25 mean? That's the predicted genotype frequency of p squared, right? Or CYCY, the genotype CYCY, in this population if evolution was not occurring. That middle number, 0.5, is the predicted genotype frequency of the heterozygous, or CYCB, if no evolution was occurring. And lastly, 0.25 is also the predicted frequency of CBCB if no evolution is occurring. So this is what we should see in our population if evolution was not happening. This is what it would look like. So what we do now, we compare this. This is our null hypothesis. If our data matches that, it means evolution is not happening. So by null hypothesis, I mean this is what the genotype frequencies should be with allele frequencies of 0.5, 0.5 if no evolution is happening in the population. So let's compare. Here were our actual values, 0.3, 0.4, and 0.3. And as you can see, they're slightly off. We predicted we should have 0.25. We actually have 0.3. We predicted we'd have 0.5. We actually have 0.4 here. And here we predicted we'd have 0.25. We actually had 0.3. So what we can see here is that our values are a little bit off from the predicted values, which means evolution is affecting this population. They are not in Hardy Weinberg equilibrium. So we can use this tool to see if evolution is actually happening to this gene, this color gene, for example. And of course, in class, we're going to look for essentially exact matches. If they match exactly, you are in equilibrium. In real population biology, you'd probably use additional statistics to see if your actual results are statistically significantly different from the results of the population. But we'll keep it simple in class. So we are going to work on some of these in lecture, and also you're going to do some of this in lab. So work through this and do some of the example problems in your textbook to get ready for lecture, and we're going to work on some of these in class on Tuesday, I guess. I'll see you then.